So first of all, a very big thank you to the organizers for having me in this meeting. It's been a great week, but with more complex organisms. So the final talk is good that we have a very simplistic organism here with not many genes to talk about and just one biological component or maximum two. So I'll talk about cytokinesis in bacteria first. And if time permits, we'll also see some amount of DNA segregation in bacteria. So what we are interested in the lab is two aspects. One is the partitioning of the genetic material itself. There's the DNA. But of course, I should confess that I don't look at the whole genetic material. I only look at plasmids, which carry a lot of important genes for the bacteria to actually survive in the environment rather than just have it replicating and dividing. And the second part is how the cytoplasm itself is partitioned and the two daughter cells arrives is the cytokinesis. So I'll talk about the second part here. And story goes like this. A long, long ago, there was a time when all the beautiful things were there in life, and there are only two kinds of organization, with all these things what you saw in the last 1 week, and the other was this. There was no kind of organization, and this was the concept of a bacterium that was put forth even as late as 1962 by Roger Stanier and von Neil. And the humble soap bubble figures again. The division was simply breakage of this bubble, and it was binary fission. The term goes there, the binary fission, how bacteria divide. And I thought at some point of time I would have to skip these slides in talks, but every conference I meet at least somebody who forces me to keep these slides even now. So bacteria do have a cytoskeleton, and they play important roles like cytokinesis, DNA separation, polarity, cell shape, whatever we can imagine in eukaryotic cell. And there's a separate class of proteins called as Walker A class of ATPases, which are termed as cytoskeletal in bacteria, and they are not seen in, the proteins are there, but they are not of cytoskeletal nature in eukaryotic cells. So they also have spatial organization, just a cartoon, that is the division ring, which is the tubulin homologue, and the actin organizes into a not so helix anymore, but it's a short patches around the circumference that makes the cell wall around wherever this protein is, and the cell wall is built in radial hoops around here, and this is the plasmid segregating protein, which may be actin, tubulin-like, and also intermediary filaments that are involved in shaping the cell. And the idea was this for a century, that bacteria don't have a cytoskeleton, despite Jacob Monard's lab, which identified the first of the cell division mutants even before the eukaryotic mutants were identified. And this is the filamentous cell that he identified mutants that would not divide in E. coli. And these are just nuclei that is there as you've seen by EM. And these were called filamenting temperature sensitive mutants because they were filamentous at a higher temperature. And nearly from after 60s and 80s, the gene itself was identified. In the 90s, they showed that it localizes to the mid-cell site and probably involved in cell division and bacteria. And it had similarity to tubulin, that it formed this polymer, and it had a GTPase activity, GTP binding, all the biochemical things. So in summary, this is what is needed for these bacteria to divide. There's a very nice movie from Sukjun's lab using a mother cell, and E. coli. The media is just flowing here, and E. coli are dividing, and as it divides, it just keeps flowing out. It looks like an amplifier music playing. It would have been nice to have a music against this background. So this is the protein, FtsZ, that forms a ring at the division site in bacteria using its polymerizing capabilities and abilities. And most of the bacterial species, including a lot of archaea, have this protein as protein FtsZ for cell division, including, and chloroplast also utilize FtsZ. And apart from this, there are a few mitochondria in ancient eukaryotes that also utilize FtsZ. And we still don't know certain bacteria, how they divide, what they use. They don't have FtsZ-like genes. They seem to divide in completely different ways that we still don't understand. And of course, eukaryote got the actin probably from an archaea that had an actin-like gene that it uses for division. So in some ways, cytokinesis, we all know, in eukaryotes is driven by this famous actomyosin ring. In bacteria, it's driven by this FtsZ, which is a tubulin-like gene. Chloroplasts also divide using this tubulin, but this only marks the division site. So dynamins localize on this chloroplast from outside, and they are responsible for constriction. So the FtsZ itself is not responsible for constriction in chloroplast. depend on dynamics, but dynamics depend on FtsZ for localization. Yes. Exactly. So only this very ancient mitochondria and red algae and few eukaryotes, very ancient eukaryotes have this FtsZ. Most mitochondria have lost FtsZ, and they don't depend, and they don't have the other genes responsible for positioning this Z ring in the middle. So in mitochondria, they seem to have lost all the bacterial genes responsible for ring position of FtsZ, even where FtsZ is present in mitochondria. But chloroplasts retain all those features. Ring is positioned by the same mechanisms like bacteria. So this is what FtsZ is. The structures really made us finally agree that yes, they are tubulin-like, and only when you see, we believe it. So only when tubulin and FtsZ were solved together, they appeared in the same journal, same issue. Tubulin has the GTP binding motif, the same GTP binding motif. The hydrolysis loop is highly conserved. So this hydrolysis loop that hydrolyzes the GTP. They form filaments, but FtsZ differs from tubulin. That tubulin has this 13 protofilaments or more filaments. FtsZ is a single protofilament of that tubulin. So it's a single subunit protofilament. That's the only difference in FtsZ and tubulin structurally. And when it comes to a cell division function, again, comparing with the eukaryotes, you definitely need these to be anchored to the membrane because it has to be coupled to the membrane. So in eukaryotes, we know that F-bar by like proteins and aniline like proteins all anchor to the membrane, link the actomyosin ring to the membrane for the constriction. Similarly, in the prokaryotes, there are proteins, at least in bacteria which are well studied like E. coli and Bacillus subtilis, there is this FtsA, which bind FtsZ to the membrane, couple the constriction force of this ring to the membrane, and that is how the cell division happens. So at the background, you see a few numbers. I must apologize as I don't know how exactly these estimates or numbers were obtained, but nonetheless, apart from those numbers, what happens is what we are interested in the lab. Yeah.
But FtsZ does need ATP?
It needs GTP.
It needs GTP.
Yes.
For its
polymerization. Yeah. So the question is, does it need GTP to assemble into this polymer structures? Yes, FtsZ is a GTP-dependent polymerization, and its hydrolysis is also dependent on polymerization. So only upon polymerization does GTP get hydrolyzed. So what we are interested in studying two aspects is assembly and constriction, but mostly I will talk about constriction because this seems to be like a highly solved problem now, but not assembly. So recent reports have suggested that peptidoglycan synthesis can drive an FtsZ treadmilling independent step of cytokinesis. So what you see here is initially there is some constriction of FtsZ, but if you block this FtsZ's treadmilling behavior with a drug here, even then the cell wall can eventually drive this cytokinesis and septation process. So you really don't need the complete dynamics of FtsZ to form the septa and the bacteria to divide. But this was a later paper. But prior to this, before that, this is also known to be true for this eukaryotic cell, which is S. pombe here, where the ring has been already assembled and it has constricted to an extent, and then you completely depolymerize the actomyosin structure there, and still somehow the cell manages to invaginate and assemble the septa, probably at a slower rate, and completes the septation. So the actomyosin ring, yes. Yeah, I'll come to that. Did I miss something? Oh, I think I.
Sorry. Is FtsZ required for the proteoglycan synthesis?
Yes.
For the cell wall integration?
Yes. So unless FtsZ is assembled into the ring-like structure there, the other cell wall synthesis genes, they do not localize there. So FtsZ is absolutely essential to get all these to that place of assembly.
But its treadmilling activity is not needed.
Is not, but again, I'll come to that contradicting result. So this is one view that the cell wall can do the septation. However, if you take the cell wall off and just take FtsZ and the FtsA protein into lipid vesicles, it seems to completely constrict the vesicles. So here you see invagination of these vesicles, and FtsZ seems to have some kind of constricting ability, and it can drive membrane constriction. And there have been several models. I think I missed somewhere, 2 slides got missing. Anyway, maybe it is there later. So So there have been several models that have been proposed for FtsZ centric view of how FtsZ can drive constriction. So one of the most famous has been, or a long-standing model has been this filament bending. Again, from EM studies or taking more from the microtubules itself, that the GTP filaments, bond filaments are straight. Hydrolysis leads to a high curvature on the filaments. And when these are bound and anchored to the membrane bending can lead to constriction, and this can drive the constriction itself. And evidence for this, more recent evidence has been coming from cryo-EM studies where they have looked at FtsZ at various stages of cell division. And even in 2 different bacteria, they see not complete rings, but arcs of rings that are associated here. In these EMs, you would not see the filament unless you go very close, but the cartoon is more helpful. So wherever there are invaginations associated, you could see these FtsZ filaments. So the model was that there is complete ring and constriction is not necessary. These filaments that are just there around the membrane here and there can bend the membrane, and then the cell wall synthesis from behind can reinforce that, and slowly over time the entire ring can constrict. So you see some asymmetric division, but over time this arcs would have moved along some other surface, and there you would have a cell wall, and you would have constructed a cell wall in that place. Yeah, so here, so going with the FtsZ centric view, 2 other reports suggested that the FtsZ treadmilling is important for laying the cell wall synthesis. And in fact, this is a model depicted here just for easy depiction, that there is a depolymerization at one end and polymerization of FtsZ at one end. What they simply did is to track the cell wall synthesis in short time. So here is one example where they first added this for 30 seconds and then this cell wall. So these are all cell wall analogs which are just fluorescently labeled. They will label the cell wall as it is synthesizing. So when you initially added this red cell wall agent, it was there. It got labeled. Then you switch to the green one, it's at the periphery. Then you switch to the blue one, that's even at the periphery. So it shows a radially inward cell wall synthesis. That is what is happening with the moving of the FtsZ filaments as it is going, and the cell wall is built from outside to inside. And when you do a shorter pulse, you can see this patches of red and green and red and green. So that again says that cell wall is made in these radial hoops, and depends, and they could show that this really depends on the treadmilling behavior of FtsZ. If you treat with a drug that block this treadmilling and stabilize these filaments, you would not see this cell wall synthesis in these radial hoops. Removal. So that has become only clear like 3-4 months back, that the polarity of the addition is opposite to that of the microtubules in FtsZ. So in the minus end of the microtubules is where FtsZ would actually get added. So that only three months back it got, with one single mutation they identified. And so this is the treadmilling model. The treadmilling drives the cell wall synthesis, and as you go, you can drive the synthesis. But there is another model for this, that the filament sliding model, equivalent to that of the actomyosin. Although there are no motors in this, it is suggested that the FtsZ filaments themselves can slide past each other, and that leads to constriction. And the evidence for that again comes from cryo-EM studies, and this answers a question, Vaishnavi, that there are at least 3 or 4 filaments width of FtsZ in a dividing cell. So you have only 3 or 4 filaments here, but the circumference, you can observe an entire ring-like structure made of these small arcs, but they go on to form a complete structure. But these may be late stages of division when the ring is there. So there are again 2 contradicting cryo-EM data that show full rings in liposomes as well as in bacteria, and again arcs and this, but this may be just differences in preparation and freezing procedures and everything.
As it constricts, then the FtsZ should turn over?
Turn over, yes. Okay. So the GTP hydrolysis will lead to depolymerization, and as it's constricting, it will turn over.
And that's observed?
Yes.
Okay.
So if 2 filaments are there together, the actomyosin with opposite polarity, as they are sliding is what constricts the membrane. So same way, this FtsZ rings, once they are assembled and they are turning over and they are sliding past each other, they decrease in diameter. So that will be the constriction. So that's the next question. So all these models impose that FtsZ have lateral contacts between the filaments. So that again, last year, evidence from Mycobacterium tuberculosis crystal structures suggests that there are at least 2 kinds of such interfaces that might contribute to these lateral interfaces. But this has been a big question, so this was happily accepted that now we have already at last found these lateral contacts between these filaments. But and again, what do these do more than studying the cell? What understanding has come, a lot of understanding has also come from simplifying the systems on planar lipids. So Mutison and Lewis first published in 2014, where they laid on a bilayer, lipid bilayer, the FtsA, which is the membrane anchor, and then to that is bound the FtsZ. So in this movies, you will see that FtsZ then starts to settle on that, and you will start seeing these vortices being formed. So these are the FtsZ filaments that you are seeing, and FtsA is on the lipid membrane bilayer, and when both are put on this lipid membrane bilayers, they kind of self-organize into these vortices-like structures, and this probably shows a more closer view of that. So you can start these vortices being formed. This is reasoning is treadmilling of FtsA itself, but what at least at 2014 when they studied FtsA and another membrane anchor that I showed you earlier was ZipA, they studied both these proteins, and these vortices were observed only with FtsA, not with ZipA. So they had suggested that it is FtsA that somehow remodels FtsZ and getting to these vortices, but a later experiment is here where they removed the FtsA and they just put the membrane part of FtsA to FtsZ directly, and there is no FtsA, it is only FtsZ with a membrane anchor, either at the C terminus or the N terminus. Both of them form these vortices. So it is completely FtsZ driven dynamics and not FtsA driven dynamics. The only difference between N terminus and C terminus was this: the direction of this movement of this vortices changed, and that is because FtsZ has an intrinsic curvature, and if N terminus is on either side, depends on orientation how FtsZ binds to the membrane. So I'll just get to that slide in a moment, that cartoon slide. So this was the suggestion that FtsA remodels FtsZ to form these vortices, but not ZipA. But ZipA might be doing additional things to FtsZ, that they are simply bundling and not allowing this FtsZ treadmilling, so that is why they did not see it. And more recent evidence suggests that, again, cryo-EM structures, the rings that you see here are FtsA themselves on lipid bilayers. So FtsA itself assembles into these ring-like structures on lipid bilayers with its membrane anchor, and over that, what you see is this FtsZ filament. So this is bottom, lipid bilayer, then FtsA. Then over this FtsA, this is the next top level, where the FtsZ filaments are running across these FtsA rings. So what is FtsA doing here is it's preventing these FtsZ filaments from coalescing together into these bundles. Yeah, this consists of, I think, 12 rings or something like that, 12 subunits of FtsA, each of these rings. So each of this is a ring of FtsA molecules, and I think there are like 12 or subunits, 12 or 18 subunits, I don't remember the exact number. But there are more subunits of FtsA in each of these rings. So this kind of finally says what FtsA might be doing, and there are more genetic evidence also to these that FtsA might actually be preventing FtsZ from coming very close together and bundling and remaining as static filaments, like what ZipA does. Okay, and this was the N-terminal C-terminal ratio. So if you had a filaments at N-terminus or the C-terminus, their movement of rotation was either clockwise or anti-clockwise, because FtsZ has this intrinsic curvature and orientation or intrinsic chirality on which side the N-terminal or the C-terminal membrane anchor, N-terminus and C-terminus of FtsZ themselves are. So on a biplanar surface, they create these vortices either clockwise or anti-clockwise. On a three-dimensional surface, so this was done by using the same constructs by just incubating them with mammalian cells.
So these are bundles of...
These are bundles of FtsZ.
The bundles of FtsZ are oriented with this, the individual filaments are oriented?
Yes, they're
parallel orientation?
They must be parallel. So that has not been still clear whether these are parallel filaments or anti-parallel filaments. But since they are moving in one direction, it must be parallel.
Or at least there are more in one
direction. More in one direction. No, that's the other interesting thing. These, at least measured by fluorescence, they are about 1 micron in diameter. All The size of filaments are again, EM shows that there are around 130 to 150 nanometers. But in this paper, they had estimated by single molecule tracking of FtsZ as long as they can see the filament, and they estimate around more than 340 nanometers here. So it can, I don't know which will be the right one. It's twice the size, but both may be right. No, the single filaments of FtsZ themselves are just this much in length. Either by EM, it's estimated to be 120 to 150 nanometers. And this paper measures by single molecule tracking, and as long as they can see before it falls off the membrane, assuming that they are passing through the entire subunit, they measured 340 nanometers. So on a mammalian cell surface, an FtsZ with an anchor at this side would just deform the surface as making these domes. But if you had a C-terminal membrane anchor, then they would create dents in this mammalian surface. So if the surface was deformable, depending on this chirality, they would have deformed the membranes itself. But here the substrate is on the glass surface, so they are just propelling and they are just rotating. And to add to all of this cell wall thing, the FtsZ Centric view to add to this one, most recently again, there has been a complete simulation of the cell wall. And what I simply understand about this is that they make a cell wall before they degrade a cell wall linkage here. So these are the cell wall linkages. The cell wall architecture and distances and everything is very well understood by electron microscopy, and the dimensions are clear, and it is 1 layer thick in E. coli. So they take a 1 layer thick cell wall here, and before they break the cell wall linkages here, they build another wall inside, and then they break these linkages, and then they iteratively go down to make the cell wall. And if they apply a constrictive force beneath this, then they can see that a force of 31 piconewtons per nanometer as unit length on this cell wall or on this membrane, you could get a reduction in the radius. And if you did not apply a constrictive force, cell wall synthesis alone did not account for a constriction. So that is all that I understood from this paper, and this was the simulation, but one needs to look into more what kind of simulations or what it was.
Just to get this straight, before the new part of the cell wall is synthesized, an old part is broken, or the other way around?
So that is, again, it needs to break down at least somewhere before the cell wall synthesis starts. So that wherever there is a crack or there is an opening, that is where new cell wall synthesis happens. So there is a small constrictive force probably does that breaking of the cell wall, or what they did is they just had few links broken, but the new cell wall layer is made completely here. So that is again not clear how much cell wall is made.
The reason for this question is I just want to understand for myself. So are you saying mechanical instability has to be created first,
yes,
and then repaired?
Yeah. So that happens in cell wall synthesis. So even in normal cell wall synthesis along the radial, this one, the bacteria are constantly hydrolyzing the cell wall. So it's not they are made and they are just left there inertly. So they keep hydrolyzing them, and wherever there is more gap created, or that is where the more cell wall synthesis again get localized. So
not intercalation.
No, it is not intercalation. It is broken, hydrolyzed, then you get more synthesis, and when they are overactive there, you just make more cell wall there.
Breaking could happen chemically, no? You can
Yes.
just hydrolyze. Yeah, exactly.
Yes. But so what I didn't understand is what this constrictive force, what does it mean for them to have this constrictive force before the cell wall is made? So, but the conclusion was that they need a constrictive force, and cell wall synthesis alone could not account for the reduction in radius unless they generated a pre-existing constrictive force before the cell wall is made.
So you mentioned that the GDP bound FtsZ is it has a curvature, intrinsic curvature.
Yes.
That curvature is smaller or larger than the curvature after
the GDP bound polymers are highly curved. It's like only 25 nanometers in diameter. But the GTP bound polymers can also be curved, but their curvature is not so much. Okay. The subunit angle is like 11 degrees or something.
And what is the curvature of the ring after it forms the ring?
The initial diameter of E. coli is like a micron in size. So that should be the size. And the vortices that are shown, their diameters match exactly 1 micron. So it might have been matched to this 1 micron, but of course there are bacteria which have larger, slightly larger diameters, where FtsZ of 1 micron curvature might be different for each different bacteria.
So in short, the constriction is not driven just by the spontaneous curvature of FtsZ.
No, that is what these different models are. So the one hypothesis was this spontaneous curvature of FtsZ driven by hydrolysis is sufficient to bend the membranes and constrict. But then this filament sliding model and other models say otherwise. And the treadmilling is another model where the treadmilling actually drives the cell wall synthesis, and that's what drives the invagination.
So this model is a purely mechanical model, isn't it?
Yes.
So that's the reason why they need a constraint. I mean, if they had a mechanics model coupled to a synthesis, chemical synthesis, then there could be other alternatives possible.
This was only
Purely mechanical.
Yeah, purely mechanical.
If it's pure mechanical, then the only way to grow a next layer is by constriction.
So I just like to just a clarification. What is the causal, sort of direction? Does the FTS ring form first before the constriction starts?
Yes.
Or is it the claim that you need a kind of a dent first and then?
No. There's no need of a dent, but there could be a need for cell wall synthesis in the region where FtsZ forms. So what happens is that you have this cylindrical cell, and normal cell wall synthesis is mediated not by FtsZ but some other molecules, and cell wall is made like this. But just before division, if this is where the ring is going to form, there is a FtsZ dependent cell wall synthesis in this middle region. And while this cell wall synthesis happens, FtsZ also coalesces into a ring-like structure. And without that, invagination will never happen. So if you take FtsZ mutants, which don't assemble the ring, even by EM, they are absolutely smooth. But if you take a mutant further downstream, after FtsZ has localized, FtsA is also localized, and then you block the cell division, what you get is slight invaginations there, suggesting that FtsZ, FtsA could have initiated the constriction or initiated the membrane invagination. The cell wall synthesis enzymes are still not there. So they both could have invaginated. That is where the FtsZ centric view started with. Yeah, that's what I said. That's why I put that numbers in the background. So probably Manoj will say how these estimates were made on this one. Okay, so then the idea was that FtsZ binds GTP, it forms these straight filaments, but the question remained like how these rings formed. So this, then I take you back nearly a decade ago. Yeah.
When does FtsA localization happen and how is that?
Immediately after FtsZ.
I thought FtsZ is held by FtsA.
True, but the ring, when you see, if you don't have FtsZ, you don't see FtsA also. You don't see the FtsA rings.
Okay, so then peptidoglycan synthesis has to occur. Some sort of
first thing to come there, FtsZ needs FtsA to bind to the membrane. So this is where FtsA is. But if you don't have this, this does not form a ring by itself. So this is what forms the ring-like structure, but it's coupled to A just to membrane anchor, and this as it forms the ring, FtsA is also on this. No, but even if you didn't have FtsA and you had FtsZ with a membrane anchor, you can get rings. I don't have FtsA, but I have a direct anchor on FtsZ to the membrane, I can get rings. So that's precisely what is seen here. So this is a multi-layered lipid vesicle, and somehow they managed this FtsZ with a fluorescent tag, with the membrane tag, inside these lipids, and FtsZ spontaneously assembled into these ring-like structures. And you can even see them in time-lapse movies. Multiple rings, when they come close together, they just coalesce into rings. So here you don't have any other proteins. It's just FtsZ with a membrane tag. No FtsA, nothing. It spontaneously assembles into ring-like structures. And at the same time, what we did is to express these bacterial FtsZ in yeast cells, and we still saw ring-like structures. So this surprisingly, again, doesn't have any membrane anchor. So even without any membrane anchor, FtsZ could spontaneously assemble into a ring-like structure. So it has an intrinsic curvature to it. Yeah, so they are like 400, 500 nanometers as we measured by fluorescence. They are smaller than what is seen on the planar membrane. Subunits can decrease. So as long as I have a longer sub, exactly. So that is probably this diameter of 400 nanometers or something. 15, it can go, invagination can somehow go to 25 nanometers with FtsZ. So what is happening as curvature increases to FtsZ, a lot of FtsZ is leaving the rings, that is for sure. So in the yeast cells, what we saw is this side is the wild type, and this side is a movie that has GTP binding as normal as the wild type, but the GTP hydrolysis is reduced like 30-fold. So this one you can see all those linear cable-like structures somehow coalescing into this ring-like structures, or here you see only patches, but well, I couldn't make much sense of this, so that is when I made all these mutants, and this 30-fold reduced hydrolysis mutant forms this single linear cable initially, and when you look in time, it forms this ring-like structure somewhere, and then this entire filament is drawn into this ring-like structure. And again, these diameters are like 500 nanometers as measured by fluorescence, but I'll show you EM of these soon.
In the lipid bilayer experiments, even in the absence of curvature, FtsZ appeared to form rings. But I'm trying to understand why, or to my mind, what appears to be a paradox, that here when you, like you mentioned earlier also, the rings form in the cytoplasm that are smaller. Is there a reason why they don't assemble on the Pombe membrane itself, or is there some special
No, I don't have any membrane anchor here. In this molecule of FtsZ, right? It's just FtsZ.
So in the in vitro experiments of
They
Martin
had a
Lowe's and
membrane
company, they had to have FtsA as an anchor, is it?
Either FtsA or the membrane anchor part alone of FtsA into FtsZ.
Okay. Yeah, sorry, I missed that.
Just one question. On the left side, on the left-hand side, it is a wild type.
Of FtsZ.
So there you are saying that ring formation occurs much faster
Yes.
than here.
Yes. So these
what-
are also like, as soon as you polymerize, you have GTP hydrolysis, depolymerization, and they also form these rings, but you don't form these long polymers. These long polymers are allowed to form because GTP hydrolysis rates are reduced. So the polymers are more stable as they are.
But eventually they also form ring.
Eventually they also form ring.
So what is the inference you are trying to draw?
So are these simply hydrolyzing GTP across the length and they suddenly have this curvature, or it was a spontaneous fluctuation here that this somehow happened to anchor to this side of the filament, and that kind of interaction with the filament drove this ring formation. We don't know how this ring formation is driven. So the ring assembly is still a question.
So the total amount of FtsE is similar in both the cases?
Total amount will be similar.
Okay. So
Expression
then
level-wise,
probably what you're seeing is smaller number of rings, but larger number of them.
Yeah.
Small sized rings. But this guy is just breaking up into two.
size-wise, yeah.
The
this is
radius.
slightly
The
larger.
radius.
This is slightly larger.
Yeah.
So when you say fold, what is the kinetic parameter? Is it Vmax or Km, or what is it? What is the kinetic parameter that is being changed here?
Okay, GTP hydrolysis is the number of GTP molecules hydrolyzed
turnover,
per FtsE
turnover.
turnover, yeah. Per FtsE molecule, that's the parameter. So, and again, like those are vortices. I don't know if these are similar to that, but you'll see this ring in the yeast cytoplasm kind of seemingly rotating. And so just as this filament is entering this ring keeps rotating, and this may be just driven by this filament that is just entering this. So as by FRAP, we can show that this part of these filaments are actually entering this ring. So this, you can see the bleach region moving closer and closer to the ring. So the filament is actually entering the ring.
Why are they forming at the ends, like the rings? Why can't it just...
No, that is simply because in this case, the filament has grown to the length of the yeast. If I had a longer yeast, the filament would have grown even longer. And ring formation, probably again, this is the free end, so that is where the ring forms. So why this did not loop over completely? Again, in a spherical cell, if I had it, maybe this would have had a chance to loop over, and this end could have connected to this. I might have got a very large ring. So I have of the same thing of a different mutants. You can have this end-to-end connection, and then nothing happens to that polymer. It just stays there because these filaments are annealed to each other ends, and they're just a loop in the entire yeast cytoplasm.
Srinivasan, this 1 micron radius of the ring, is it something intrinsic to the, does it happen when you only do it on a cell surface, or even with a planar membrane you see rings of similar sizes?
That is 1 micron. Even on the planar surface with the lipid anchor,
it is always close to 1 micron.
There has been some interesting hypothesis to that did the FtsZ's intrinsic curvature restrict the bacterial cell size rather than anything else? And example for this is this huge bacterium which is called as Euplopezium, which grows in the gut of a fish. So this is around 400 to 600 microns in length. And what happens in the fish is that as the fish is eating, the bacteria grow inside and grows up huge. And when division has to happen, this has a lot of DNA. It's polyploidy. When division happens, FtsZ ring forms here. Euplopezium, I don't know to spell, is EP. Easier is to Google like Esther Angert, and she's the one who found with Losig this bacterium. So the FtsZ ring assembles at the ends of this. It divides into a single cell inside this mother cell. And the diameter of these rings is not more than 1.52 microns, even in this bacterium. And after the cell divides, it eats up the entire mother, all the DNA nutrients, and this cell grows on to become big, and eventually you get 2 big cells inside this, and the mother cell lyses. So they grow as the fish is growing in the morning and feeding. At night they divide. So that has something that did FtsZ ring itself restrict the size? More so because analysis of FtsZ sequences has shown that it is one of these primordial 7 proteins that existed on the planet. Even before the codon became a triplet code, FtsZ is supposed to have existed. So again, what we did is, if this is the protofilament axis where FtsZ subunits are stacked on one on top of other, the mutation that I showed you was this one, the GTP hydrolysis mutant. And then we made a bunch of mutants already known from the E. coli literature on this surface or this surface. And one of these mutants formed only these linear filaments and never formed rings. It never went on to form this ring structure that I showed you. So this kind of led us to propose at that time that we still don't know how ring assembles, but GTP binding forms linear polymers, GTP hydrolysis drives curvature, and then in addition to that, there are lateral contacts between these FtsZ filaments that can stabilize the ring structure. But then what are these lateral contacts? So the questions are then, how are these rings assembled, whether these filaments are parallel, anti-parallel, but this, although EMs have been revealed now, but whether they're parallel or anti-parallel is still not clear. And the exact nature of this so-called lateral contacts, which remained a question, but I just showed you the MTB paper that they have identified some residues which may be lateral contacts. And how is this force generated in the absence of contraction? That seemed to be like a largely solved problem with all this treadmilling and all these models and everything, which Manoj may talk more. And how is the final closure of the septum? Because as I said, the curvature problem. So as you keep increasing this curvature as you are decreasing, you can probably get to around 25 nanometers or 12 nanometers in size. But after that, how do you close the septum? And there is evidence that when FtsZ has completely left the ring and you can no more see FtsZ at the middle, cytoplasm is still diffusive. So you can bleach GFP on one side, you can get recovery on the other side of the daughter cell. So there is probably a hole which is closed a little more slower than.
It force without lateral contacts. Is treadmilling by itself sufficient to generate force without lateral contacts?
So whether that requires the cell wall synthesis, then you from the FtsZ is required to drive cell wall synthesis, then cell wall just invaginates and that kind of thing, but then it doesn't generate constrictive force. I don't know if treadmilling alone will be able to generate constrictive force or it is just driving cell wall synthesis. And this one? Oh, let's have 3 questions. A lot of animations, sorry. Yeah, so the 4th generation, the absence of motors, right? So if the treadmill, but the treadmilling could also lead to this sliding mechanism. So essentially, if treadmilling is leading to this sliding across these filaments and leads to sliding, then it will be the treadmilling leading to sliding of these filaments and changing the intrafilament contacts. It will need lateral contacts in that sense. Yeah.
Are these lateral contacts required for anything other than the ring formation? I mean, what happens?
So that's a related question, that if lateral contacts are not there, can you still have constriction? So if the filament sliding model and the treadmilling model is true, then do you essentially need lateral contacts? So I think that lateral contacts will also be needed for contraction of the ring, and that treadmilling and lateral contact, constant changing of these lateral contacts will lead to the constriction. And incursion of the
just have
cell wall synthesis,
cell wall synthesis
yes.
and that should yeah.
Single protofilaments of FtsZ could simply treadmill and make the cell wall, and that goes back to the EM where only arcs are seen and you don't have a complete ring-like structure. So again, both might be existent, both mechanisms may be acting in this cell wall synthesis and construction. So then the question was, how are these rings assembled in vivo? Because we don't know much about the ring assembly. So what we did, okay, so before I get to that, this is the EM of the ring, the massive ring with the GTP hydrolysis mutant that I showed you. So you're looking through sections in yeast itself, and each of these black filaments is the 4 nanometer thick FtsZ filament. And at some point you must have noticed a tail somewhere here, which is that spooling ring that was coming into this EM. So we did this to see if we can also see the wild type structures by EM, by tomography and everything. But unfortunately, we did not see any wild type rings in the yeast. It was just lost in the contrast, never got there. But this mutant, we could see a very nice tight rings. And on the next slide, you can see one section of this, which are each of these black protofilaments are FtsZ ring. And here what I have done is, it's not a perfect model, but I've just drawn green lines over all the sections wherever I see these green rings. So this is the final picture that you may get of this entire thick ring structure, and just rotating 90 degrees here. So this is a very thick filament. It's almost like 600, it's a barrel, 600 nanometers, and inner diameter is around 250 nanometers, and outer diameter is again 400 nanometers.
So this is from data, from the electron.
Yes. This is from these sections. So if I play this movie, this is through these sections.
So when you have the spooling in yeast, you're sort of generating a...
This is what it generated.
I'm sort of still trying to think, are you building up the width of the ring, or are you actually...
No, sorry, if I can borrow this, I always have this.
Z, like when you have a hose pipe and you...
Yeah, this is, initially this is what you saw in that green structure. Then I just go over this, and if I have many of it, it's Hanuman's tail, just build up.
Whose hand is it?
Whose hand? Oh. Yeah, that we don't know. Where is the hand? So this again, they agreed to, in a meeting in this time, they had modeled this like automated segmentation. We want to see if we can get filament lengths and see if we can build some kind of model out of that. What is the interfilament length? What is the filament length, and how they are structured? But it didn't go that far. But still, this is a nice picture to see finally in a three-dimensional view what the ring looks like. And if I were to just take the top slice of this with 2 or 3 filaments, that is what this EM picture in 2014 that was shown. So that is what might have been the wild type. We didn't see any wild type in the EM because it was just lost in the contrast. Or we did plastic embedding, maybe cryo would help. So what we did later is, no, wild type is also, out of that barrel, if I had just taken the top slice, that's what would have been the wild type. So if this is the barrel, I just take the top slice of 2-3 filaments.
I understand. My question was a little bit, do you think you have a possible explanation why this is, I think, a slower GTPase mutant, right? That's, yeah. Why do you think that the slow GTPase activity results in this higher, thicker barrel?
That's where so the FtsZ has only this kind of, it can go from this curvature to this curvature. So it cannot have much higher curvature or much broader. So it is the intrinsic curvature of FtsZ filament.
And you're saying that they aggregate by these lateral interactions? Is that why they...
The lateral interactions are between these filaments, is what is the presumption. And to find if there are such lateral... Yeah. Yes,
sir. Sorry.
You may have said this. So the cell wall synthesis machinery also accumulates around the FtsZ, or is that?
Yes. So the cell wall synthesis, so FtsZ, FtsA.
FtsA is
the anchor. And once this comes, there is the next cell wall, the transpeptidoglycan and the transpeptidase.
They are associated with the
FTS. They will associate with this and they will form the cell wall across. The main activity is outside, but they are linked into the cytoplasm with these proteins.
So wherever there is cell wall synthesis, there is FtsZ over there.
Wherever there is FtsZ in the septum, there is cell wall synthesis. Otherwise, FtsZ is not doing the cell wall synthesis. It's only the septal cell wall synthesis that is. So what we did is took this yeast system to identify mutants that do not assemble into rings, because this was easy to do in yeast and I would not have been able to do it in bacteria, because any mutants that don't assemble into rings, I will not get back those bacteria anyway. So we found several mutants, and 2 of the mutants were interesting, which is what he is working on, Ajay, my PhD student, and 2 of those mutants are this L68 and T127. They form these nice helix-like structures. You can see this. So what I thought at that time is this, what we think is this helix is something, if I had this and just pulled it apart, it's not a compressed ring-like structure, and if FtsZ itself is a compressible spring-like thing. So that's an idea, and these are the lateral contacts, defects between these, and that is what these mutations are. So these might be lateral contacts or ring assembly, I exactly don't know, but what we started doing is to take these mutants, put them back in bacteria, and fortunately or unfortunately, whatever, I can take it either way. These mutants, when I don't have this tag, they function very well in E. coli, meaning that they give rise to colonies. I don't know if there are any defects in cell length or not yet, but when I have these same mutants with this tag, the wild type is perfectly functional, but not these mutants. That already tells me that probably there are some defects in these mutants with respect to the filament architecture, and therefore they are not functional. So in a way it is good, but then I cannot image them in live, these mutants alone, so it's bad in that sense. So what we at least know so far now with these mutants is that if I express these mutants in a wild type background where the wild type FtsZ is there, these rings do not go into the ring very efficiently. So if you look at these, the wild type, I'm staining with immunofluorescence, that is the antibodies to that, and I'm just looking at the fluorescence of these mutants with the green fluorescence. So in case if it is wild type and wild type, I almost get an equal ratio of these rings. But as soon as I get to these mutants, this mutant is fine, I get almost 50%, but this particular mutant, I have a large number of wild type rings in the cells, but this mutant, I don't see a lot of green fluorescence. So they are not going into the ring efficiently. So probably these mutants are defective in ring assembly, and they also have a slower GTP hydrolysis inferred from this FRAP analysis in the yeast expression system. So they have a slower hydrolysis than that of the wild type. Wild type turns around with 10 to 11 seconds, but these mutants are around 20 to 25 seconds. So implying that the turnover rate of these mutants also might be affected, but again, not in bacteria. checked only in the yeast expression system. We need to do this in bacteria as well. But it shouldn't change much. It's all intrinsic to FtsZs. And of course, there are cell length defects. So this is the wild type expression. The wild type is always there. It's not that we are removing the wild type in this case. That we haven't still done that. We can remove the wild type and check what the cell lengths are. So these mutants have very long filaments, and a large number of cells end up being very long. And there are few in wild type that doesn't happen when you overexpress wild type. So this is really overexpression of these. They can perturb the division. And these two are proteins which are responsible for bundling of FtsZ. So the idea was that if these mutants are more sensitive when bundling of FtsZ is not happening. So again, you see a large number of longer cells when you express these mutants rather than wild type. Wild type, in fact, reduces the cell length of these mutants, brings back the cell length to the normal cell length, but these mutants are unable to do that. They have a large number of longer filament cells. So there is some defect, but we exactly don't know what these are doing. So idea is that they either have a lateral contact interface defect, and that is why they are not compressing into a ring-like structure, assembling into a ring-like structure. Or these mutants could also be restricted in this domain motion, which again recently the solving of crystal structures finally came to a solution like what is actually causing this polymerization? What is the conformational change? How does a single protofilament can have cooperativity in its polymerization and also have treadmilling? So what these crystal structures showed that there is an open conformation and there is a rotation of this domain by 27 degrees when the polymerization can happen, and when GTP, it is not linked to GTP hydrolysis, because even with GTP bound form, you can get this close conformation. So polymerization and the ability to polymerize is not linked to hydrolysis switch in this case. It is simply, we don't know what it is linked to. It is polymerization itself driving this open conformation. So if you have a GTP bound molecule, it can remain in this close conformation, but a switch to this open, as it is binding the pre-existing polymer, it exists in an open conformation, and all monomers in the polymer will be in the open conformation. And that is associated also with this central helix moving to some extent and restricting that movement of the helix. So these mutations also could have affected this domain motion, and therefore you end up with a filament that is slightly more twisted than the intrinsic helicity or chirality in FtsZ. So filament length, this L68W interestingly has been already studied. We got it in random mutagenesis screen, but there have been a site-directed mutagenesis, and EM lengths show that those are slightly more longer than, they are like 200 nanometers compared to the wild type. So the filament length is definitely, the individual filament length is affected. So that might also have triggered this. So that brings to the idea that if the filament length is longer, is the stiffness or bending rigidity of these filaments are also affected, and that is why it is taking this helical shape. So that collaborating with, I don't know where Debashish is. So Debashish is trying to see if we can extract this bending modulus or filament stiffness from these yeast filament movies with just fluctuations. With extracting the fluctuations there, if we can estimate some bending rigidity and filament. So that might also have contributed this helical shape, and he's also trying to see if, how do you get from filament to a ring an intermediate helix? Can you get that by changing filament stiffness and putting in a lateral contact interaction? And so that kinds of brings to the end of my cytokinesis part. If time permits, we'll go to DNA segregation, but I'd like to thank Cloud and EMBL facility where I could do the EM sometime back. And also thanks to Ranjit and Sanjita Labs. So we three are independent labs, but we share our resources together and we are a single lab unit. So I really like to thank them, because without them, it would not have been possible to pool resources and actually do all this work. And all the PhD students and graduate students who have, 2 have left the lab now and 2 more might come in, and all the funding, DAE, of course, for its most generous funding. We have intramural funding and we didn't have to worry about writing grants, but nonetheless, DBT and DBT, we did get grants and microscopes and other things from those grants. So thanks for all the generous support from the funding agencies. And thank you. And welcome to Mysore. So, okay.